Welcome friends to a new session on genomics and proteomics. In this session, we will discuss mapping populations. According to Mendel, the development of phenotypic characters is governed by hypothetical factors now called genes, and their alternative forms, which are known as alleles, segregate independently. Sutton and Boveri proposed the chromosomal theory of inheritance in 1902, soon after Mendel's findings in 1900. According to chromosomal theory of inheritance, the genes are located on chromosomes. Morgan provided the first evidence for chromosomal theory of inheritance in 1910. He demonstrated the inheritance pattern of white eye gene of Drosophila melanogaster located on the X chromosome. Morgan described the essential features of linkage between genes in 1911, and Sturtevant published the first linkage map of Drosophila melanogaster in the year 1913. In many species, morphological markers are used to construct linkage maps. These markers will space considerable distance between each other. Geneticists searched for more abundant markers and protein polymorphisms, and these were the first molecular variations used to generate linkage maps. But the environmental influence and limited number of polymorphisms were the major drawbacks, and these led to the development of DNA markers. RFLP, or restriction fragment length polymorphism, was the first molecular marker used in linkage mapping. Later, RFLP is replaced by PCR, or polymerase chain reaction based markers, and SNPs, that means the single nucleotide polymorphisms. The use of DNA markers allowed the construction of linkage maps of many plants and enabling the mapping of quantitative trait loci, or QTL. Construction of linkage maps requires a suitable marker system, an appropriate mapping population, and software for the proper analysis of the data. Mapping populations. Population is used for mapping the genes is commonly called as mapping populations. In other words, population is suitable for the linkage mapping of genetic markers is called mapping populations. Mapping population usually obtained from control cross or crossing of two or more genetically diverse lines. Mapping populations are usually used for determining the distance between pairs of loci or genes and to map them specific locations in the genome. They also help in the identification of the molecular markers linked to the genes or loci of interest. Such markers can be used for the marker assisted selection, or MAS, for the genes of interest. Thus, mapping populations serve as the basic tool for the identification of the QTLs and its effects. First step for the production of mapping population is the selection of parents. The parents used for developing of mapping population must differ in their DNA sequence and phenotypic levels at maximum extent. The variation in the DNA sequence level is essential to find the polymorphic molecular marker.